for you. And yes, it's recording. The other, so this DNA extraction is the only thing we're going to be doing live today. The other videos were ones that we did at the beginning of the stay-at-home order. On Sunday morning, Bernice and I, we were told that campus was closing on Monday. Bernice and I came in on Sunday morning and put together 3 videos on that I've shared with you in that handout. One is on PCR, the second one on gels. I think that would be the only 2. Yes, only 2 that I shared with you. And so you'll have access to those on YouTube, and you can view those as you wish. We can't, it'd be very difficult to film Bernice pouring a gel today and maintain 6 feet. And also, I am not a very good, I have no practice at handheld camera work, so it would be quite, everybody be seasick before we're done. But just fitting since most of our fish come from the ocean, but hey. Anyway, any questions? Any more questions? Okay. Doing things like, oh, yes. Jasmine asks if you could request partners. That's fine with me. Send it in an email. Send me an email. You should have it. I'll also give you my email now in the chat. If you have a partner request, I will honor those. Sure. Oh, I need to send that to everyone. Again, let me also echo what Dr. White said, as I'm really excited that so many of you showed up this morning to participate in our sequencing success. It's actually a better showing than we had last year when we were live. I think I had 12 students last year for this event, when we were hoping to have over 70, but today we have 29 or 28 participants, and also a visitor from a community college. I'm blinking on its name now. I don't know, Anjali, do you want to say anything while we're waiting? Oh, because I haven't sent it yet. So, but I think it's really exciting that you guys want to do things like this to learn more about science and the process of science. And there is, if you're planning on applying to UCR, in the application, there is a place to mention that you did participate in this type of an event that was offered by us on campus. I don't remember what that's called, but it's something like additional supporting things to help your application, okay? And definitely take advantage of the cross-enrollment, like Dr. White said. We're paying RCC prices for a UCR class. It's actually a well-kept secret that this is possible. Very few students take advantage of it. And that also comes with some peer mentoring as well. So the UCR student will help you kind of navigate through. Hopefully, I think this will take place either winter or spring, and we'll be back on campus, and the student will help you navigate the campus and find your classes, help you with getting around, getting involved at UCR. Okay. And if we were all in person, just 20 minutes would go like by in like 10 seconds because everybody gets off out of sync, and it takes a while. But it is a pretty exciting day. We have 3 labs. You kind of saw the lab as I spun the camera around the mess because we had to hide a bunch of stuff. There, unfortunately, a lot of thefts on campus as things were closed down. So we had to hide all of our and lock up all of our equipment, and so the lab is a mess. We have 3 labs that look pretty much the same, and we would have over 70 students working on this project. And normally I don't do any of the talking. I have other people, and I just float between the rooms and manage everything. So I haven't run this in a while. We do this also in the fall for all local community colleges, and we have so that means we've been doing this now twice in the summers. I don't think we're going to do it virtually this year, though. That would be, I don't know, we'll see. Okay, any questions? Okay. The next few steps, incubations are shorter. So lastly, let me tell you a little bit about our labs. So we are in the Neil A. Campbell Science Learning Laboratory. And if you used Campbell Biology, that is who it is named for. And Neil Campbell was a graduate student here. He got his PhD in botany. And he taught at San Bernardino Valley College, as well as UCR at the end of his life while he wrote his book. And after he died at an early age, his wife, Rochelle Campbell, started donating money to UCR because that was Neil's passion, was supporting undergraduates. So she donated the money for the renovations for the laboratory, and she named it after him. And then, wow, 2 years ago now, the building was named for Rochelle and Allison Campbell. So Neil's wife and daughter. So, and as far as we know, it's the only building on campus named for two women, and the only building in the college named for women. So we have a special building that we are mostly occupied just in the Campbell labs. There's one other research lab upstairs, but other than that, we occupy the whole building. So it's a very nice place to be. This is the longest I've been here, and no, that's not true. I had to teach from here one time because my internet went out. Well, we cut the internet cable while trimming some bushes, so, you know. Anyway. Okay, how many people per group? Good question. I haven't looked yet. Since we have, I think, 28. I think 7 groups of 4 would be good. Or maybe, that would be a lot of presentations. I'll let you know. It's going to be around 3 to 4 to 5 groups. I don't want them too small. I don't want them too big because then it's just not enough work to set around. If they're too small, there are a lot of presentations, and if we give 20 minutes per group, it could go quite long when we come back together. And just to remind you, we will return on July the 24th, same time, for presentations, and those we'll talk more about in the following 4 weeks, or the weeks in between. Okay, about time. Okay, it's time to move on to the next steps, which is potassium acetate. So what's going to happen next is I'll flip you up to Bernice. She is going to add potassium acetate, which we abbreviate KOAC, and mute me. Bernice, there you go. So Bernice.
Okay, hi. So I'm going to take the sample out of the bead bath, and then I am going to add the 5 molar potassium acetate. It's going to be 225 microliters. Okay. Okay. And now we're going to invert the 2. And so what potassium acetate will do is it's going to precipitate the proteins and the SDS. So after inverting, we're going to place the tube in ice for 5 minutes. And we wait again.
Yes. So that's exactly correct. So the potassium acetate causes the SDS to precipitate and pull down proteins, lipids, anything that's got positive and negative charges. So that can come out of solution as a white precipitate, and the ice just helps make that go quicker. And the next step will be to put it in the centrifuge and spin that down. Oh, you're going to need the balance too.
I have it.
You do? Okay. Spin that down to pellet the SDS crud, as we like to call it. And we will then proceed with the extraction. So at this point, the DNA is in solution. Okay, the what we will be precipitating right now is the stuff we don't want, and then the next precipitation will be the DNA. Okay. Any questions while we wait on the precipitation to take place? I think the air conditioning finally came on. It was quite warm in here this morning, and it's finally cooling down. Okay, so okay, oh good, we're getting some questions. So one question is, how big are the labs at UCR? They're around 24 to 30. The labs are, the Campbell labs, what we teach, are 24. Most of the labs, the major labs, the rooms can hold only 24 students. So, oh, okay, more good questions. There was another question about, microliters. Why are we using microliters? That's a good question. Mainly because we don't need a lot of DNA to for sequencing. Also, enzymes that we use for the next step that I'll tell you about in a minute can be expensive, and so we want to minimize the use, how much we use. Those are the main reasons. It's the volumes that we need. And it is small. I mean, 1,000 microliters is 1 milliliter. Okay, so we're working in very small volumes. And then, can you get a copy of the procedure? Yes, it's actually in the handout that I put in the chat. It's the procedure she's using for the DNA extraction. And then what does SDS stand for? It stands for sodium dodecyl sulfate. I'll write it in the chat. So sodium dodecyl sulfate is the proper name for another, for a compound that if you look at your shampoo bottle, and unless you use the really, really, really expensive fancy stuff, the main detergent or soap and shampoo is sodium lauryl sulfate. It's the same thing, okay? It's just that we use it in pure form, and the shampoo, of course, has all other kinds of stuff in it. But the main component of the shampoo is water and sodium dodecyl sulfate. Okay, time for the next step. Yep.
Oh.
Yeah, you're I muted you.
Okay, so we're gonna take the tube and centrifuge it. I'm not sure if you can see the centrifuge, but we're just going to spin down the protein. I have a balance tube so that the centrifuge is balanced and it does not break when it is spinning down. But so really all we're going to do is have the protein settle down to the bottom of the tube, which is everything that we do not want in cell debris. Settle to the bottom of the tube as it spins for 5 minutes at 14,000 RPM. So as I mentioned, the tubes have to be placed directly opposite of each other to keep the centrifuge balanced, and then we put a lid on tight so the tubes do not fly around, and close the lid and start. This will be for 5 minutes.
Okay. Let me mute this in case it's a little loud. There. Okay. So the question was, do you need to find the materials to do this? And the answer is no. We're just doing a demonstration, and you will get the you'll get results that we have done in the past. So this is just for you to watch and to hopefully one day get to do it in a laboratory, but not for this exercise. Okay, so the centrifuge, like Bernice said, what's happening is the solids are pelleting. They'll form what's called a pellet. Let's see, hold on, I will go to the whiteboard. Okay, so what we mean by pellet, in case you haven't done this before, is that we have our tube here. Okay, this is our what's called a 1.5 mil tube. Okay, we have our liquid in it, and
we can't see you. We can't see
centrifuge
you.
is complete. We're going to have all the solids
We
down here
can't
at the
see you.
bottom. Okay, and this is called our pellet. The liquid here we call supernatant. Okay, so Bernice is going to take, so right now the DNA is floating around in the supernatant, and that is what we're going to take to the next step. So she's going to remove the liquid from this tube, filter it to get rid of the last of the solids, and then we'll have DNA in a separate tube and leave the pellet in this tube. Okay, and that's, then we will pellet the DNA by adding isopropanol, and you probably will not be able to see this pellet because they tend to be really, really small. And if it's a good prep, almost clear. So we will see. And of course, I didn't switch the cameras. Sorry about that. Let me show you my picture. There. Okay, so now you can see the pellet and the supernatant. So the pellet is the part down here, and the supernatant is the liquid that's left over with the DNA floating around in it. Okay. And let me reset the camera. Yeah, that's correct. You don't need to acquire any of the fish samples. You're going to be given some that were processed last year. Okay, should be about ready. Okay. So you should hear the centrifuge come down momentarily, I think. And we'll go to Bernice then. Okay, any questions in the chat? Nope. Okay, good. Okay, there we go. The centrifuge is breaking. So we'll go back to Bernice for the next couple of steps. So when you're in lab, you always want to be listening to your centrifuge, and as soon as it stops, you want to be standing there to take your samples out.
Okay, so as you can see, the protein and all the cell debris did pellet. I'm not sure if you can see it, actually, but
Sort of see it, but yeah. It's not going to focus on the. Okay. But yes, you can see there's a nice white pellet and a good bit of liquid.
So now I'm going to transfer the supernatant, which has the DNA, into a separate tube, and I'm going to be using America cloth as a filter so that we don't catch any of the debris that we do not want in the second tube. So all I'm doing is I'm folding a square of America cloth into a triangle and then a second triangle. And then through one side, I'm going to pour the supernatant into the second tube. And this is just going to filter the DNA. And so it's going to be 700 microliters of the DNA that's going to be transferred. So here's the tube with the debris and the DNA. We're going to take the DNA out. If you can see that. And I'm trying to avoid the pellet as much as I can. So you take 700 microliters and then into a second tube. It's going to be hard to see. You filter through.
Great. So you can see this requires some dexterity to get this done. So this step is just in any floating solids in the liquid we want to remove because those would interfere with final steps.
And so now I'm going to be adding 600 microliters of isopropanol. And as Dr. Burnett said, this is going to be to precipitate DNA out of solution, and we will have a pellet after spinning the tube down.
Right. Yes. So the isopropanol is also known as rubbing alcohol, but this is at 100% isopropanol, and it changes the hydrophobicity of the solution, so the DNA will come out as a potassium salt. So we're getting the potassium salt of the DNA, and we will then do another centrifuge step. But this time, the pellet will be what we want. The supernatant will be discarded.
I'm really quickly going to make a balance just so that I can spin the tube.
Okay. Yes, these centrifuges have to be balanced properly, or else a lot of them these days won't spin if they're not balanced properly, but it will ruin the motor if they're not balanced correctly. And so we have another 5-minute spin. So as you can see, we're using chemistry to isolate DNA. There also will be RNA in this pellet, but that's okay. It won't interfere with anything that we want to do with this sample. But we have been first mechanically disrupted the cells, and that's usually what differs. So if this were human cells, we wouldn't have to hardly do anything. If it was just like cheek cells, you just put them in water and they lyse open and release the contents. tissues might need some mechanical disruption. Plants are often a lot more fun because you usually require liquid nitrogen to freeze the cells, the tissue really, really well, and then it breaks open because it's so brittle. You use a mortar and pestle. So students and scientists as well like playing with the liquid nitrogen. So that adds a little fun component. But after that, once you've broken open the cells, the DNA chemistry is the same, and so the steps you use and the reagents are identical, whether it's human or plant or bacteria. Okay. And we're doing what's a fully manual prep, meaning we're doing every step and not using anything, but there are kits that are available you can buy, and I have one here. You can buy these kits, and instead of using hulls, they use these columns. So they use filters that bind to the DNA, and then you wash this with much of the same chemicals, but and reagents, but the DNA binds to the column rather than being a pellet. These give much cleaner preps than what we're doing today, but they cost more money, and they don't need that quality of DNA, so we're not using it. We're just using a. Oh, good question. So the question is, what happens if you don't pull out samples from the centrifuge immediately? What happens is your pellet softens, right? So it's sitting there, and these pellets are pretty sticky, so they stick to the plastic of the tube. And I just realized. So this is the tube that Bernice has been using, or like it, and the pellets will stick to the wall here, and you can just actually just dump the liquid out, is what she'll do in this step, and then maybe pipette off the last drop. But if you let the tube sit in the centrifuge, that stickiness to the wall is going to decrease, and if you dump it out, you might dump out your pellet, and that's not good. So it's best to always be ready at the centrifuge for the next step and get it out as soon as possible. Also, in a classroom setting, somebody was probably waiting on that centrifuge. So it's best, and even in regular labs, they may not have that many, so you don't want to take up a centrifuge just by letting their samples sit. But that's why you always have an ear out for when the centrifuge starts breaking, so that you're ready when the lid opens. It should be about done, I think. I don't know. 2 more minutes. Okay. And if you go work in a lab, the steps are going to be almost identical. The times that are done for the centrifuging, for the ice, for the heat, will all be different. There's nothing magic about them. We've optimized this protocol so it goes as fast as possible. If Bernice were doing it by herself, she'd probably complete it in less than 30 minutes and probably 10 samples in less than 30 minutes. Well, let's say 45. With students, the first time they process a sample, it usually takes an hour and a half, just because you're learning how to do everything and manipulate all these tubes. But we've optimized it so that it works well in a 3-hour class time. But other labs might do things quicker, they might do them slower. You just have to learn the protocol that you're given in that particular setting. A lot more activity in the hallway than I would have expected.
Yeah.
So well, the centrifuge should be about done, so I'll wait. But It is possible, especially at UCR. A lot of students do research in laboratories. Bernice worked in a laboratory that studied neurospora for most of her time, but she was an undergrad, and even now, before the shutdown, she was working on finishing her project before she goes off to grad school. But it is common for students to do research on campus with professors here at UCR. And in fact, my group encourages it quite a bit. I did undergrad research, but that was at the University of Georgia a long time ago. Okay, so now she should have a pellet. Keep see it.
No. Yeah.
Okay. Good. So that means it's a really clean pellet. So in this case, DNA, of course, has no color.
Can they see me?
I don't think they'll be able to. Let me see.
Oh no, are they?
No, you're right. Yes, yes, yes. There, sorry. So there you go. Now we're back with Bernice. So there's a tiny, tiny pellet there. Okay, yep. So, and believe it or not, it will stick to that tube. So a good DNA pellet is like, they often call them glassy looking, it's shiny, should be clear, colorless, and it takes some practice to learn to be able to see these pellets if they're clean enough. Okay, and she's inspecting it to make sure she gets all the isopropanol off, because that would not be good. And so what's the next step?
We're gonna add ethanol, ice cold ethanol. It's going to be 500 microliters. And so all I did was remove the supernatants, which we do not want in this second step. We just want the pellet, which has the DNA. So 500 microliters of ethanol.
Right. So the ethanol will remove the last of the isopropanol. And this is 70%, so there's some water in it, which will help remove any extra salts that we don't want. So this is a wash step. So we're just washing this pellet. So she'll just flick it around a bit to get the pellet moving, but we're not going to disrupt it. And there's still enough ethanol or alcohol in the solution that the pellet will stay as a pellet. It will not, as a solid, it will not go into solution.
Okay, now we spin again.
5 minutes. 5 minute spin, and then you're approaching done.
Yeah.
So this is actually the last step of the protocol to clean it, to wash it off with the ethanol. We will then pour off the ethanol, dry it in the air, just leave the tube open, let it air dry to remove the last of the ethanol, because that's bad stuff for the amplification step. The ethanol would denature any proteins, which would include the polymerase that we need for the amplification. So, and then we will resuspend that pellet in TE or water today. So we're going to be using a solution called TE, which has tris and EDTA. So tris is a buffer. It'll buffer it around pH 8 to protect the DNA. And the EDTA will remove magnesium mostly from solution so that enzymes can't degrade the DNA. And then once we finish the last step, or once she pours off the ethanol, I'll talk with you about the amplification. While that happens, I'm going to erase the board. I'll be back. Okay, yes. Towards the end, I'll talk more about the presentation right before we break up. Uh, okay, so was the tube placed on ice for 5 minutes? Was that the annealing step? No, that was to precipitate the SDS, and that will to remove protein and lipid from the solution. The annealing step is what we'll talk about next. That's in the PCR or the amplification. Okay, now, so the centrifuge should be finishing up soon. We will, so remind you that the gene that we're going to amplify is actually off the mitochondrial DNA. And to remind you, right, the mitochondria does have its own genome, just like chloroplasts have their own genome. And the mitochondria of animals is pretty simplified, the genome. But the gene that we're going to amplify is the 16S rRNA gene, right? So 16S rRNA is one of the RNAs found in the mitochondrial ribosome, one of two. And there is a gene that codes for that RNA. And typically we think of genes as coding for protein because the majority of them probably do. And that's mostly what we focus on in biology class, are the genes that code proteins. But there are genes that don't encode proteins. In fact, they encode RNAs. And there's a whole bunch of classes of RNAs that are required for a cell to function. And one is in the ribosome. And it's a really good gene because, as you can imagine, this gene is not evolved very quickly because the ribosome is a complicated structure and it's so central to the functioning of the cell, it cannot evolve rapidly. So that means that it's very stable between species, within a species. We'll come back to that because the centrifuge just finished. Okay.
So as before, I'm just going to pour out the ethanol. And then using a P20, I'm just going to take out any remaining ethanol, being careful that I avoid the pellet.
Yep. Yes, we worked too hard to lose the pellet at this point.
Okay, and since we don't want any ethanol in our DNA, we're going to leave the lid open and keep it in the bead bath so that the ethanol evaporates.
Exactly. So, and then in a few minutes, we will add the TE solution that will complete the preparation of the DNA. Okay. So let's move on then and discuss a little bit about the next step. Okay. What? Why is it? Yeah, that's correct. Okay, sorry. I was thinking that it looked flipped. Okay, so to amplify the 16S rRNA, okay, like I said, it doesn't evolve very fast, and that's very good because one, we're going to need to amplify. So when we do this with students, we have potentially 60 to 70 different fish samples, different species. But we don't want to set up a PCR with, we'll see in a moment, we need these things called primers. We only want to use one pair of primers. We don't want to have primers for each fish, because that's the point. We don't know what the fish is, correct? So people realize that if I take the 16S gene from a bunch of different fish, so this is the sequence of one, just shown as a line. This is the sequence of another. So these are different fish samples, 1, 2, 3, 4, etc. Okay, I have a whole bunch of fish, and you look at the sequence, right? So I'm just showing the sequence as a line. What the scientists realized is that there are regions of the 16S gene that are the same. They're identical between species, okay? And even between genera, or between the genera of the different fish, these are sequences are identical, okay? And then also happen at another side, okay? These are identical. These are important for the function of the 16S rRNA gene, okay? In between is called the spacer region, and this sequence varies, okay? So that means that we have conserved regions for the primary sites, and then we have a variable region that is unique to the species, okay? And that's how this works. So to amplify a gene, what we need to do is replicate it, right? It's just simple replication, but not in a cell, it's in a test tube. And it's actually simpler than in the cell. If you've studied replication, in the test tube, it's only leading strand. Okay, we don't have lagging strand replication going on here. So it's actually really straightforward. So to do this, we're going to use a technique called PCR, or the polymerase chain reaction, okay, or PCR. Okay, and in that handout I gave you, there's a nice animation that I like that you can, that will show you how this goes in detail. But for PCR or for replication, we need a few components, okay? And those components are common for a lot of things. So we need a polymerase, of course. Okay, and the one that we are using, we'll be using in the research is called TAQ. Okay, TAQ. So this is our polymerase, DNA. Polymerase, of course. We need nucleotides, right? So the ATP, these GTP, CTP, and DTP. Okay, and we abbreviate this DNTP. Okay, so we need the DNTPs are the building blocks of the DNA, the A, C's, G's, and C's. The last critical thing that we need are primers. Okay, because this polymerase cannot make DNA on its own. It can't just start making DNA. It has to have a primer site. So we usually show these as arrows. Actually, I do have other colors. So, okay, we need one arrow that will prime one strand. and we need an arrow or a primer, excuse me, that will prime on the other strand. And these go in these common regions that have identical sequence. So this way it can prime. Maybe this is tuna, maybe salmon, rockfish. Swai is usually a very common fish we get in the class. Tilapia. They can prime across all of these genera because the sequence has not evolved or changed at all. Okay, so what we can do then is from the 20, 30,000 genes that are found in a fish, we can amplify just this one because these primers bind only to this region of the 16S or this region of 16S. And from that, we then go through the steps of the polymerase chain reaction to generate our results. And the steps are very straightforward. So step one, we have to denature the DNA. So we mix all this together in a tube, right, with our sample, like Bernice just prepared. And then that goes into a machine called a thermocycler. And I just realized they're all locked up. So I can't show you one, but you'll see a video that we made and you'll see the thermocyclers. Okay, so we mix all this together and we put them into a thermocycler, or also called a PCR machine. And what that machine does is heats and cools, and that's it. That's all the machine does. Because step one, we want to heat to 95 degrees Celsius, or sometimes even hotter, 98. So very hot, okay, almost boiling. Okay, and what's going on at this step? Does anybody know? What goes on? What do you think is happening to the DNA when we heat it to 95 to 98 degrees?
The DNA strands are separating.
Either speak up or type it in the chat. 95, 98 degrees, what do you think is happening to the DNA strands? Exactly, the DNA is separating. So we call that denaturation, right? So when we denature the DNA, remember, DNA is double-stranded, okay, and held together. So this is one strand, forward strand, reverse strand, and it's held together by hydrogen bonds between the bases, right? So this could be an A, and this would be a T, so you get 2 hydrogen bonds, and G and C, 3 hydrogen bonds. So the denaturing is breaking that bond so that they come apart. And that allows the primers to be able to bind. Okay, so the primers can come in and bind so that you have a priming site now. Okay, so that's denaturation. Yep, okay. Next, we lower the temperature to around 60 degrees Celsius. Okay, and that's annealing. So that's when these primers come in and they're able to form hydrogen bonds. Okay, so that's called annealing. And then lastly, we raise it to 72 degrees Celsius and new DNA is made. Okay, the polymerase can come in, bind, start at the priming site, and add bases to that primer. So we're extending the primer, and that step is called extension. Okay, so denaturing, annealing, extension. And then we just repeat that around 30 times. Okay, so around 30 cycles, and we have a nice amount of 16S RNA gene that we can then analyze further. Okay, so that's the PCR. This was invented in the mid-80s, and it really changed the way molecular biology was done. PCR is so central to almost any research project on campus. So take some time and watch the video that I gave you. It's impossible to draw what's going on. I tried the very first time when I first started teaching. I tried to draw on the board the steps as we go through step 1 and then cycle 2 and cycle 3 and cycle 4. It was a disaster. So I only use the animation, and I don't want to sit and bore everybody to death while we watch it right now. So, but take some time and study the animation. It's probably older than you guys are, but it really, really is good. And then there's others. You can look up a really good resource for animations is the Dolan DNA Learning Center. I know the people who make these videos, and they are very scientifically accurate, but very accessible. So look up Dolan DNA Learning Center whenever you want to learn about PCR, CRISPR, all kinds of stuff, mitosis, meiosis. This group makes really good videos. Now, and they have one on, it's very simple, and it's narrated, it's subtitled, so take a look at it to really understand what's going on during these different steps of PCR. And then if you have more questions, when we get back together over the next couple of weeks, I'd be happy to answer questions or send me an email, and I will be happy to answer your questions there as well. Okay, so is there any burning questions right now about PCR? Not in the chat anyway. I think the next time I do this, I'm going to have a huge screen because I can't read this text that far away. Okay. I think that's all for PCR, unless there's questions. So that was, I erased my steps, so extracted DNA. We've amplified the gene using this PCR method. Okay. And that takes about an hour and a half, and that's why it will take us up to lunchtime. We get this thing running, the machine's going. We usually have a really nice lunch catered, and then by the time we're done eating and pictures are taken, and we'd come back and we would run a gel. Okay. So the agarose gel, we're not going to show you today. Again, like I said, we can't do it and be socially distant and all the what we need to be practicing. So I'm going to describe it a little bit on the board. There is a video that Bernice doing every step of making a gel, and also a video of Bernice setting up a PCR samples. So please check those out. Those are on YouTube. And again, if you have questions, please just send me an email, something by email or by when we get together next time. Okay. So the point is, we've done the PCR, but PCR can fail easily for many, many reasons. It's picky, it's finicky. So we want to make sure it works before we go on to sequencing, because sequencing is more expensive than actual PCR. So we want to make sure that we're giving good sample so that we good sequence, okay? Science is just like anything, crap in and crap out, okay? You don't want to provide bad samples. And the facility is not going to check them for you because it's not their project. So what we do is we make an agarose gel, okay? And these are in our lab about this big, and they are about the size of an iPhone, right? And they are made of agarose, okay? And so agarose is a sugar. It comes from seaweed. So the YouTube channel is, it's my personal, it's my UCR account, but click one of the links in the handout under the PCR or gel step, and you'll get to that page. If not, I will email it to everyone when I send the email out later today. So, so agarose is a sugar, comes from the giant seaweed that we see off of California. And we melt it into a buffer, and it forms a nice gel. It looks a lot like Jell-O, okay, a little thicker, but Jell-O, of course, is a protein. In this case, we're using agarose, which is a sugar, okay? And when we form the gel, we put what's called a cone in it to create wells. Okay, so we now have our gel like this, and it has wells in them. Okay, so these go about halfway down. So if we looked at the gel on edge, right, the wells would be something like that. So we have these wells, and this is where we load our sample. Okay, one of them is going to be a ladder. Okay, the ladder is something we buy. And it is our standard. So we load our ladder here, and it just so happens that most of our solutions are red when we do this. So there, we've loaded our ladder. Then we would load our PCR samples. Okay, and then we usually also have one sample that doesn't have DNA in it, so we have a negative control. Water, no DNA, or negative, negative control. Okay, so we've loaded it up, and then we put an electric current. And so we put this gel into a liquid solution, same buffer that we used to make it. We put an electrical charge across it. So it's negative to positive. Okay. And the DNA is going to migrate to the positive pole. Okay. So we say run to the red. So the DNA is going to separate out by size. The bigger the piece, the smaller the piece, the further it travels. So this would be really big, and this would be very small. The bands themselves are the same size. Okay. But the DNA inside these bands are bigger. So maybe this is 4,000 base pairs. Okay, and this would be 40 base pairs. Okay, so the smaller you are, the faster you go towards the positive pole. Now, if we have a really clean sample, we should get one band representing our RNA gene in each of these except that one. So we'll run it out, we'll get one band except the last lane because there was no DNA there, right? And if there were DNA there, what do you think would be the problem? Anybody want to guess? So if we got this result, right, so what we do is we set up PCR for these samples, we add DNA, different ones, so this might be rockfish and trout and salmon and catfish. And then one sample, though, doesn't get DNA. It gets a little bit of extra water. So, yes, this is called gel electrophoresis. So what do you think would be, how would we interpret a result if we got a band in this lane? Anybody want to guess? Or does anybody know? Okay, so if we got this, that means that there's something wrong with the reagents, right? That there was some contamination, and that's not good. So how do we know what these bands, did they really come from rockfish, or is it some other contaminant that accidentally got into solutions? And this happens, okay? We do get contamination, it's very easy. So we always run the water control to make sure that if there's no reaction here, these samples amplified from the DNA we put in the tube, okay? So that's very critical. So in order to do this, to see the DNA, we add ethidium bromide, which is an orangish colored dye, but we put very little. The ethidium bromide accumulates in the DNA bands, and we put ultraviolet light on it, and that causes the bands to light up. And so we take a picture with this under ultraviolet light. And I was going to show you some results, but I can't share my screen, so I won't be able to this morning. But I will send an example of results in the email when I send it later. And this was actually will be from last summer with RCC students. So, okay, so questions about PCR, agarose gel electrophoresis? Do you want to clean up to go?
Do I plus that? It's still
Hmm?
It's still
Okay. Whatever you need to do. Hang out. Yeah. Okay. So anyway, Bernice has an appointment later, so sorry, we were discussing. Any questions come up? Not yet. Okay. So like I said, these are very commonly done things in lab. We isolate some DNA. We have a particular gene we want to amplify. We use PCR to do that. And all we change really is the primers, right? If we want the rest of the mix is almost always the same. The primers are what select the gene that we're interested in. So maybe it's like when we do the nematodes, we use a different gene because the nematodes are evolving very rapidly. So we use a nuclear gene rather than a mitochondrial gene because we need something that's evolving more rapidly than this particular gene. Maybe I want to clone a specific gene. I just change my primers, and that's that. Okay. Very common first steps to anything, and then we always check on a gel to make sure that it looks nice and clean. Okay. And the gels I'm going to show you did turn out quite nice, and so we got a common single band. So next thing we have to do is sequence. Okay, and sequencing is very similar to PCR. At least the type of sequencing that we're using. It's very similar to PCR. So we need to remove the polymerase, we need to remove the nucleotides and these primers, okay? They all have to go from the remaining sample. So we have our tube now with our DNA sample. Maybe there's 50 microliters. We took out 10 microliters to run on the gel, so we still have 40 microliters left. So we're gonna clean up this remaining DNA by the remaining PCR sample. There's various methods to do, and it's not important which method that we use. Whatever happens, remove the primers, the nucleotides, and leftover polymerase. Okay, then we now have a nice clean DNA band or sample that can be sequenced. And we give it to them. We add a new primer. Sometimes it's the same primer, but only one primer. We split this into two tubes, two different primers sometimes, or one. And then we give it to a guy named Clay over in another building who prepares the sequencing reactions and runs it on a machine. And if you're interested, you can do a Google search for Sanger sequencing. If you want to learn more about the process, SEQUEN. Sanger sequencing, this is the type that we're using. So if you want to watch videos on Sanger sequencing, Dole has good ones. Other groups have pretty good videos on sequencing. I don't typically go into it a lot because it is so similar to PCR. A lot of times students confuse the two, okay? And so I focus on the PCR, but if you want to go further and learn about Sanger, definitely go for it. Some self-directed learning is always good. So then it takes about, depending on how many samples and what's going on, it takes Clay anywhere from 24 to 72 hours to get us our results back. And the results come back, and I will provide a short video on how to analyze those results during the week, next week. I didn't have time this week to prepare that. So I will provide that with you another time at a later date, and work with you on your next steps. Okay, any questions so far? I know that's a lot, but you have time to digest it and go through, watch the different videos that I've provided links to, and send me questions, or we'll probably be getting back together again in a Zoom conference in your individual groups to talk about sequence analysis and results. And so definitely write down questions to ask me then. Okay. Any other questions?
Do you want me to show them the TE?
Oh, sure. Okay, so Bernice is ready to do the last step. Let me turn the camera back on her. And
Okay, so Okay, so the ethanol evaporated, and I'm just going to add 50 microliters of TE, or resuspension buffer. And this is just to keep your DNA stored for long periods of time. And so after this, you can then go on to PCR, which then follows what Dr. Burnett said, gel electrophoresis, and then sequencing. So it's 50 microliters already set. And that is about it. You do want to shake it to make sure that your DNA is in solution. But you can store this in -20 for years, and it's good to use. And that is the last bit of DNA extraction.
Okay, so yes, that's the whole process. Then we would, in lab, typically we prepare what's called a master mix for the students, and they will add their DNA to that master mix, and we turn the machine, this thermocyclers on, and there we go. Turn the thermocyclers on, and we are back in business. So that would make so that there would be time for the machines to amplify the samples. Okay. So that's the process. Any questions? Oh, somebody's waiting. No question. Okay. So what's going to happen next is either this afternoon or Monday, I will send you an email with your particular fish sample that I will assign to you fairly randomly. And then and there will be instructions on how from that information to find out what to find the sequence file, which will be in a Google Drive. And then from that, you will have a video to watch to show you how to do the analysis. And yes, I can include the handout in that email as well. And then it'll also assign your groups, probably do that Monday, and get so that you guys can get started. The analysis doesn't take long at all. I think we usually do this over several weeks because students also have classes and work. So they just probably only need to meet a couple of times over the next few weeks. First to do the sequence analysis, and I can be available. And I actually have a student who did this a couple of years ago who has volunteered to mentor a group or two. So I will assign you to either me or Kimberly, but I'll check in with everyone also. And answer any questions you might have about the sequence or the analysis. Finally, then after that, you'll write an abstract. And an abstract is a very short, usually no more than 250 words, summary of your project. So you would start with a couple of sentences about introduction. Why is it important to identify fish? Why is it, whatever, what question you're addressing, then a sentence on the technique or two, a sentence or two on the results, and then on the interpretation of the results. So it's basically a summary of your project in 250 words or less. And then I will give you feedback on those abstracts. And then you will work together as a group to write that abstract, and then also work together as a group for your presentation. And presentations, I'm going to say 15 minutes will be plenty of time. I would use Google Slides, so it'll be easy to share your screen. What we'll do is, and I'll give you more detailed guidelines on the number of slides I think you should have, what information should be on those slides. And then, like I said, like 15 minutes, each of you should take turns presenting. One of you can show your screen and the others can talk and asked for the slides to be changed. I did this in the last quarter for my class for their projects, and it worked really well. So if you use Google Slides, not only is it free, but it's easy to share, work together, collaborate remotely, and also then share them over Zoom on July the 24th, I believe it is. So like I said, 15 minutes, we will have each group go. Order can be determined at that time. And that will wrap up the sequencing to success for this summer. Any questions? So be on the lookout for an email from me. It'll be either Jim Burnett or James Burnett. I don't know how it shows up. It seems to be random. So just be on the lookout for an email from me and with your fish and the next steps. Okay, questions? Okay, Bernice, do you want to say any last things to them?
My own.
You're not muted. I can. Okay, Bernice.
Hi. So I hope you enjoyed the experience. DG is always great. So if you ever come to UC Riverside, I would recommend you trying to be involved in the program. You can either be a student and take a course, or you could volunteer in their club. The staff here are great, and my experience with them for the past 5 years has been phenomenal. So yeah, thank you for participating, and I hope you enjoyed this.
Yep, great, thanks. So yes, Bernice has been a very valuable member for the last, as you said, 5 years or so. She's going off to UC Berkeley in the fall. We're going to miss her, but off to a new adventure in grad school. Please feel free to email questions to me. If you have questions, if you want to ask Bernice something about her time here at UCR, send me an email. I'll forward it to her. If you want to ask her a question about that. Okay. So, well, I hope this was an interesting morning. I was trying to do some new things with cameras. It seemed to go pretty well. Thanks for bearing with me. First time doing something like this live. And as everybody, this is all, every day is a first these days. I learned a few things and how to improve for teaching in the fall. And also I hope you guys learned a lot on PCR, barcoding, and molecular techniques such as PCR, gel electrophoresis, and definitely DNA extractions. All right. Anybody have any comments or questions for me? Great. Well, I think that's all for this morning. I planned for 3 hours just because I didn't know how long this was going to take. About an hour and 45 minutes. That's not bad. I wasn't sure until this morning how all we would do and how far we would get. So yep. So this recording will be made available. It won't be available. I won't get the link for a couple of hours, but it'll be in the email that I send to follow up with this either this afternoon, but most likely Monday morning when I have time to sit down and make all the groups. And definitely send me, I know 2 people have already sent me email requesting partners. If you have a partner that you want, just send me an email. I'll be happy to do that. Okay, Well, if that's, if that, we're done. I will hope you have a good weekend. Enjoy the weekend. It's gonna be pretty warm, but that's okay. Get out, do something, get some exercise. And hopefully we'll see everybody back here on July the 24th. And at least once or twice, if not in between. Okay. All right. That's all. Bye, guys.